The genetic code, the fundamental language of life, is composed of sequences of 3 nucleotides called codons, each corresponding to a specific amino acid or a stop signal during protein synthesis. Despite the redundancy in the genetic code with multiple codons encoding the same amino acid, not all synonymous codons are used equally across organisms. This phenomenon, known as codon bias, has been extensively studied in the context of translation efficiency, protein expression levels, and cellular fitness. Among the factors influencing codon bias, codon optimality plays a crucial role in determining how effectively a ribosome can translate mRNA into proteins. This article explores the concept of codon optimality and its profound impact on ribosome decoding efficiency, drawing on extensive evidence from empirical studies, evolutionary perspectives, and computational models. Codon optimality refers to the differential efficiency with which ribosomes decode synonymous codons. Some codons, termed optimal codons, are translated faster and more accurately due to their higher affinity for cognate tRNAs, while others, called non-optimal codons, are translated at a slower rate. The degree of codon optimality is influenced by factors such as the abundance of tRNAs, their charging efficiency, and their competition for the ribosomal A site. Studies in various organisms, including Escherichia coli, Saccharomyces cerevisiae, and mammals, have demonstrated that codon optimality significantly affects translational kinetics and the folding of nascent polypeptides. The link between tRNA availability and codon optimality is evident in genome-wide analyses. In E. coli, highly expressed genes preferentially use codons that match abundant tRNAs, ensuring rapid and efficient translation. A similar pattern has been observed in S. cerevisiae, where ribosome profiling data reveal that transcripts enriched with optimal codons exhibit higher ribosome density and faster elongation rates. Conversely, genes with non-optimal codons often show increased ribosome pausing, leading to slower translation and, in some cases, translational errors. A seminal study by Garden et al. 2014 in S. cerevisiae demonstrated that codon optimality governs ribosome elongation speed by modulating codon-tRNA interactions. Their ribosome footprinting experiments revealed that ribosomes tend to stall at non-optimal codons, leading to ribosome queuing and altered translational dynamics. Additionally, a study in Drosophila melanogaster by Presniak et al. 2015 further underscored the impact of codon optimality on mRNA stability, showing that transcripts enriched in optimal codons exhibit longer half-lives compared to those with a high fraction of non-optimal codons. A particularly compelling example of codon-dependent translational efficiency is observed in stress response genes. In Saccharomyces cerevisiae, stress-induced genes often contain non-optimal codons, which serve as a regulatory mechanism to fine-tune protein synthesis under fluctuating environmental conditions. This strategy prevents excessive energy expenditure during stress, while ensuring that proteins crucial for survival are synthesized when needed. Similarly, in mammalian cells, synonymous mutations that introduce non-optimal codons into housekeeping genes have been linked to diseases such as neurodegeneration and cancer, emphasizing the biological significance of codon usage patterns. The evolutionary pressure shaping codon usage has been a subject of extensive investigation. Highly expressed genes in both prokaryotes and eukaryotes display strong preferences for optimal codons, suggesting that natural selection favors sequences that maximize translation efficiency. Comparative genomics analyses reveal that rapidly growing organisms such as bacteria and yeast exhibit stronger codon bias toward optimal codons than slow-growing or multicellular organisms, likely due to the selective advantage of efficient protein production in high-growth environments. Intriguingly, codon optimization strategies differ between taxa. For example, in Escherichia coli, selection for translation speed is evident, as genes involved in protein synthesis exhibit a high frequency of optimal codons corresponding to the most abundant tRNAs. In contrast, In mammals, codon usage is influenced by a complex interplay of factors, including GC content, mRNA stability, and translational accuracy. This divergence underscores the adaptive significance of codon optimization strategies tailored to an organism's ecological niche and metabolic demands. Beyond translation speed, codon optimality has far-reaching implications for protein folding, co-translational assembly, and cellular homeostasis. A striking example of this is observed in protein domains requiring precise folding kinetics. Studies have shown that slow-translating non-optimal codons, strategically placed at critical junctions of protein sequences, can enhance proper folding by allowing sufficient time for the nascent polypeptide to adopt its correct conformation. This mechanism is evident in ribosome-associated chaperone systems, which assist in the proper folding of proteins synthesized with complex structural features. Moreover, codon optimality plays a role in synchronizing ribosome movement with mRNA decay mechanisms. A landmark study by Mishima and Tomori in 2016 in zebrafish embryos demonstrated that transcripts enriched in optimal codons are more stable, whereas those with non-optimal codons are preferentially targeted for degradation by exonucleases. This finding highlights the interconnectedness of translation and mRNA turnover, where codon usage serves as a regulatory code influencing transcript longevity. The principles of codon optimality have significant applications in synthetic biology and biotechnology, where optimizing codon usage is a key strategy for enhancing heterologous protein expression. In recombinant protein production, adjusting codon compositions to match host tRNA availability can dramatically improve yield and functionality. For example, the expression of human insulin in Escherichia coli was significantly enhanced by replacing rare codons with host-preferred alternatives, thereby minimizing ribosome stalling and improving protein folding. Codon optimization is also critical in vaccine development. The production of viral proteins for vaccine formulations often involves codon re-engineering strategies to maximize expression levels in eukaryotic or bacterial hosts. A prime example is the development of the SARS-CoV-2 mRNA vaccines, where codon optimization was employed to ensure robust translation of the spike protein antigen, enhancing immunogenicity and vaccine efficacy. Such applications underscore the utility of codon engineering in advancing medical and industrial biotechnology. Despite the wealth of knowledge on codon optimality, several challenges remain in fully deciphering its regulatory roles. One major hurdle is the context-dependent nature of codon effects, where interactions between codon usage, RNA secondary structures, and ribosomal pausing complicate predictions of translational efficiency. Additionally, emerging evidence suggests that codon optimality influences processes beyond translation, such as ribosome recycling, RNA localization, and non-coding RNA functions, warranting further exploration. Future research directions include the development of high-resolution ribosome profiling techniques to capture real-time translation dynamics with unprecedented precision. Additionally, advances in computational modeling and machine learning approaches will likely enhance our ability to predict the impact of codon usage on gene expression. Understanding how codon optimality intersects with cellular physiology may open new avenues for designing synthetic gene circuits, optimizing therapeutic protein production, and unraveling the evolutionary forces shaping the genetic code. Codon optimality represents a fundamental yet intricate layer of gene regulation that influences ribosome decoding efficiency, protein synthesis rates, and cellular homeostasis. Empirical studies spanning multiple domains of life consistently demonstrate that optimal codons enhance translation speed and accuracy, while non-optimal codons play regulatory roles in stress responses, mRNA stability, and protein folding. The evolutionary conservation of codon usage biases highlights its adaptive significance, shaping organismal fitness and metabolic efficiency. With implications ranging from synthetic biology to disease research, continued investigations into codon optimality will undoubtedly deepen our understanding of molecular biology and provide innovative solutions for biotechnology and medicine.